Gibberellin, Ga. Gibberellic acid, also called gibberellin A3 or Ga3, is a hormone found in plants and fungi. Its chemical formula is C19H22O6. It was first identified in the pathogenic fungus Gibberella fujikuroi, which causes a disease in rice called foolish seedling. By producing large quantities of Ga, the plants become long and slender, incapable of supporting their weight, chlorotic and partially infertile. Further research established gibberellin as a hormone that is essential for many developmental processes in plants. Among them are seed germination, organ elongation and expansion through cell growth, trichome development, transition from vegetative to reproductive growth, and flower, seed, and fruit development. Gibberellin manipulation in agriculture is a common practice. The best-known contribution of gibberellin manipulations to agriculture is the introduction of dwarfing alleles into staple crops. This resulted in one of the cornerstones of the so-called Green Revolution, an increase in global wheat and rice yields. Movement. Gibberellin moves inside the plant in both acro and basipetal directions, though it is more efficient in the former case. Gibberellin movement is essential for several developmental processes. Production. They are produced in the chloroplast of the plant cell in an inactive form and activated in the endoplasmic reticulum. Gibberellin regulates cell division and elongation. Shoot elongation in this case results from both cell division and cell elongation. When applied in low concentrations to a bush or dwarf bean, the stem begins to grow rapidly. The length of the internodes becomes so great that the plant becomes indistinguishable from climbing or pole beans. Gibberellin seems to overcome the genetic limitations in many dwarf varieties. Internodes elongation is the most pronounced effect of gibberellins on plant growth. In many plants such as dwarf peas and madge, the genetic dwarfism can be overcome. They stimulate stem and hypocortical growth, positively regulate cell division and elongation, hypocortical and stem growth, and have a positive effect on leaf size and root meristem size. They affect leaf size and root meristem size. They are involved in seed germination. Gibberellins can both stimulate seed germination and suppress it at high temperatures. They are involved in flowering and flower growth. They are also key regulators of reproductive organ formation and development and the ripening of fruit and viable seeds. They are involved in leaf and fruit decay. They play a role in seed dormancy. Some seeds that are sensitive to light such as tobacco and lettuce exhibit poor germination in the absence of sunlight. Germination begins rapidly if the seeds are exposed to the sunlight. If the seeds are treated with gibberellic acid, the light requirement can be overcome. Gibberellin are used to increase crop production and fruit size. They are used to delay senescence in fruit to extend its marketing period. They are used to open flower buds. The buds formed in autumn stay dormant until next spring. This dormancy can be overcome by treating them with gibberellin. Gibberellin has been considered helpful in the removal of the harmful impacts of salt stress in rice plants grown under saline conditions. Prakash and Pratap Senan, 1990. Gibberellin-deficient rice mutants exhibited higher tolerance to drought and lodging resistance in experimental conditions. Furthermore, The addition of a gibberellin biosynthesis inhibitor led to a decrease in sucrose content and an increase in fructan content in shallot, Allium cepa. They are used to lengthen sugarcane stems to increase yield. They are used to promote early seed production and hasten maturity in young conifers. They are used to promote bolting and flowering, such that plants produce seeds earlier in beet and cabbages. In urban areas, GA antagonists are sometimes applied to trees under power lines to control growth and reduce the frequency of pruning.